Glycolysis. Okay, let's keep track of some glycolysis accounting. Okay, we're accountants, biological accountants. Glycolysis itself occurs in the cytosol. We have not entered the mitochondria yet. Okay, we only get 2 ATPs. Okay, 2 ATPs, glycolysis. We haven't entered the mitochondria, right? Then we need some pyruvate to enter the Krebs cycle. But the Krebs cycle also only gives us 2 ATPs. All of that, remember, the whole thing is to make it to the electron transport chain, which is our hydroelectric situation that's going to make us the 26 ATPs. Okay, so all of this is to get to the electron transport chain. Okay, electron carriers. These are NADH and FADH2. You can call them electron carriers. That is the whole point of this, is to make the NADH and the FADH2 to move the hydrogen ions opposite of their concentration gradient to run down and power the electron transport chain. Okay, there is a gradient. Remember, the carriers drop the electrons at the electron transport chain, and as the electrons move against their concentration gradient, brought to you by your electron carriers and some ATP, they go against their concentration gradient, up and along, and then down the hydroelectric pump, the spinner, the generator that makes our ATP. And what else makes this possible is it's like a traffic jam. Your electrons transfer, right? Your transfer down, down, and you have to have someone to accept. Come here, little electron. I love you. I accept you. Come on, come out of the chain. Because if oxygen isn't there to be the final electron acceptor, the whole thing backs up. It's all backed up. It's like a traffic backup clogged situation. And then we have to do something else and we do not make as much ATP. Let's look at it. Okay. Everything we've done, glycolysis, the Krebs cycle, is all to make these electron carriers. Those electron carriers move our hydrogen against its concentration gradient. Now, also, we are not in the cytosol. We are in the intermembrane space of the mitochondria. This is the matrix. This is the intermembrane space. We are moving our hydrogen ions. Look at all these guys against their gradient. The whole point of this is to move them here so that we can make the ATP. Okay, this is our generator. We've moved all these hydrogen ions up here to run the generator like a dam. Okay, so we get that. We're running it through. Now, what do we need oxygen for? Why are we even breathing? Who cares? We do this. Here's our electron on its little way, right? Here it is, moved all of its hydrogen, moved its last hydrogen ion up against its gradient, and there's an oxygen. Come on, buddy, right? If this doesn't happen, if there's no oxygen here, this electron stays and backs the whole thing up. This doesn't work. These don't work. The whole traffic jam is backed up. Let me see if I can, well, we won't get to that. Okay, I just want you to visualize this system has got to work like this. You've got to have oxygen here. These hydrogen ions are against their concentration gradient, and they're pumping this generator that's going to make us 36 ATPs. Here is a different view of that. Just remember, this is the intermembrane space. Why I don't always like this picture is because I want you to understand that this is the intermembrane space in your mitochondria. Okay, here's, look, NADH and FADH, right? These are our electron carriers. They're shooting our hydrogen ions against their concentration gradient to run the generator. Okay, they move here, here, and you've got to have this oxygen here to collect them, or they back the system up. Now, you can fill this out. We have how many ATPs do we get? Here we get 36. You're not going to get any water until your electron transport chain, but you can go in and do that, and I did it in class, so you can find that video if you'd like. Or you can just come here and look. Here they are. So glycolysis, you get 2 ATPs. Right? Citric acid cycle, you get 2 ATPs. Electron transport, 34. So you can go back up here, 2 to 32 to 36. Okay? See? You can do it from here. Okay, what else do we have? 2 electron transport carriers, 2 electron carriers, 8 electron carriers. Okay? And as we move on, you can do CO2 and H2O. Okay? But we're just looking at what each one of these things does. Your glucose to pyruvic acid, your pyruvic acid to acetyl-CoA. Your acetyl-CoA is your ticket on the citric acid cycle. You can't get on the ride. Now, also remember that your one glucose molecule makes 2 pyruvic acids. So your 2 pyruvic acids also make 2 acetyl-CoAs. So you get 2 tickets to the citric acid cycle. Okay, you're riding the ride twice. The number of ATP generated by cellular respiration is 30 to 36 glucose. It varies by how efficiently your NADH can enter your mitochondria. So cellular respiration in general is 34% of the energy coming from glucose to ATP. Okay, now what did I say here? I'm going to show you this. Here is what we did in class. Okay, looks exactly like the other one. Glucose, and here's our accounting as well. Glucose molecule, let's keep track. There are 6 carbons. Down here, you have 3 carbons because you have 2 of them. Remember, you get to ride everything twice. From this, you get 2 ATPs, 2 electron carriers. Okay, it takes 10 enzymes, and this is happening in the cytoplasm. You made pyruvate. You made 2 of them, though. Okay, so your 2 pyruvates come down here and make 2 acetyl-CoAs. Okay, and you get 2 CO2s from this process. So 2 CO2s from pyruvate to acetyl-CoA, 4 CO2s from citric acid cycle. Put that in your chart. You also get 2 electron carriers here, 2 electron carriers here, 8 electron carriers here. You get 2 ATPs here and 2 ATPs here. But mostly the magic happens in the electron transport chain where you get 36. Okay? So all of this is just to fuel the electron transport chain. Yes? Now, remember what I said. If you don't have any oxygen to accept these electrons, all of these electrons, we're going to back this whole thing up. It's going to be like a traffic jam on 270, right? There's no oxygen here. So it backs this up all the way. It backs this up and all the way here. Like the last thing you can do is get 2 ATPs from glycolysis. It can't even make pyruvate. It has to make lactic acid or fermentation. So you only get 2 ATPs. Okay, because this whole thing is backed up. Why is it backed up? Because these electron carriers are not able to, their passengers are not able to get off. So you have all these Ubers and no one else can get an Uber because the passengers aren't getting out of the Ubers that they're in. Okay, the only way this works is if these carriers are empty, and they're not because there was no oxygen there to take them. That's why, seriously, breathing, super important. Here's your anaerobic pathway, okay? So here's your glucose molecule. We make ATP, fine. We are trying to make pyruvate, but it's not going to work because we can make pyruvate, but we can't move into acetyl-CoA. So at this point, you can either make lactate or ethanol, and that gives off CO2. And that CO2 is what rises your bread and makes the bubblies when you are fermenting alcohol. Fermentation does not use the electron transport chain or the Krebs cycle because it can't, because those electron carriers are busy because they still have passengers. It simply recycles the NADH from here, from glycolysis, and you can make 2 ATPs over and over again. Okay, same thing here. Glycolysis goes this way. This is lactic acid fermentation, and this is lactic acid in your muscles, but there's some question about whether that's really what makes you sore. And here is my attempt at a flow chart as to how all of your digestion works. Okay, let's briefly talk about passive versus facilitated diffusion. This should be a review from everything else, but I just want to specifically point out what we have already learned from glut, Siggelt, how things are getting into the capillaries. Okay, Passive diffusion moves downhill from a high concentration to a low concentration. This is just basic diffusion. Should have learned it last semester, probably learned it right. This is like stinky smell, and it, you know, everybody gets to be part of it. This is you put a drop of water in a beaker and it's going to diffuse, right? It's just moving, passive diffusion. It's moving from a high concentration to a low concentration. No ATP is needed for this, right? Just magic. Facilitated diffusion, this is like SIGLT. You need a buddy, you need a friend. It's for larger molecules like glucose, usually uses channel proteins like tunnels or carrier proteins. Okay, so you got to have somebody moving you along. Okay, so passive is just, it goes right in there, right? Here, you got to have a channel protein or something because you're too fat. You're too fat to just go in here, or you're not a lipid. Active transport. Okay, so this is our friend and yours. My favorite is, of course, the sodium-potassium pump. You know how much I love the sodium-potassium pump. This is active transport because it costs you something. Okay, it's going against the concentration gradient. The whole point of the electron transport chain is to push our hydrogen ions against the concentration gradient. The entire point of the of the sodium-potassium pump is to move things against the concentration gradient. And what do both things do? They cost us money, and by money, I mean ATP. The currency of your cell is ATP. And so you have to use the sodium-potassium pump, which charges you ATP to get your 3 sodiums out and your 2 potassiums in. Okay? Moves things against the concentration gradient, but it costs you ATP. Also, let's not forget the exocytosis, or the just barfing out. Just, it spits it out. Puh, right? Exocytosis. Vesicles fuse with your plasma membrane and then just like barf you out. It's awesome. It's just like, yeah, it's my favorite. You get a vesicle here, vesicle fuses with your plasma membrane and it just barfs out things like insulin. Okay, a reminder again about your pancreas, right? You've got bulk transport and cell signaling. Now, paracrine is local. Paracrine is like, hey, it's like talking to your friend. Hey, dude. It's really talking to the adjacent cell. This is right here. Hey, you need to like release the bile. Okay. Also, let's not forget autocrine is a self-receptor, right? It's like setting a timer on your own phone. This is telling your neighbor something. And this is just dumping the signal into your bloodstream. Like, hey, here's some insulin. Here's whatever else hormone that we can come up with. And we're just going to dump that in the blood and see how that goes. Okay? So endocrine is a long distance hormone. Paracrine is a signal just right next door. Okay? And autocrine is itself. So here again is an overview. Autocrine is talking to itself. Sometimes it can do this across gap junctions. Paracrine is a cell to a nearby cell. This is your pancreas to your duodenum for your bile and your liver, all right there. And then endocrine is in the bloodstream. This is pancreas when it releases insulin, right? Your beta cells. Okay, again, here it is. Endocrine, your islets of Langerhans. It's fun every time. They secrete hormone, insulin, well, and glucagon, but insulin directly into your blood. Right? Just here you go. Good luck, insulin. See you. Right? That's an endocrine gland. Right? That's your beta cells. Your exocrine cells secrete digestive enzymes right into your pancreatic duct, right into your duodenum. That's why your pancreas is so cool. Okay, I'm going to make one more video and it's going to be about lipids and proteins.